And thanks for the introduction. And I'm Zang from Mie University in Japan. Today, I'm very glad to be invited as a speaker of this online meeting. Today, I would like to talk about the zebrafish model for studying the prevention of obesity. So, this is the outline of my presentation. I will talk about what is zebrafish, their introduction of the overweight and obesity, and how to develop an obese model using zebrafish lab and adult zebrafish, and how to use these models for anti-obesity screening of natural products. At first, why we use zebrafish as a disease model? The zebrafish is named for the horizontal stripes, the blue stripes running the length of its body, which are reminiscent of a zebra's stripes. Zebrafish is a tropical freshwater fish, which is originally found in eastern India's Ganges River and native to the Himalayan region. And the characteristics of the zebrafish include high fecundity and small size. For example, one pair of adult zebrafish can provide over 300 embryos every week. And the length of the adult zebrafish is around 3 to 5 centimeter, and we just need a comparable small space to house them. And the zebrafish embryo is optically transparency during early development, which is a benefit for high resolution imaging of live animals. The embryonic development of zebrafish is very rapid, and the laboratory strains of zebrafish can hatch around 3 days post fertilization. The generation time is 2 to 4 months, and the lifespan is 3 to 5 years. These characteristics make zebrafish very good for the ontogenesis study and lifestyle-related disease studies. And the genetic and organic similarity of the zebrafish between zebrafish and human is highly resolved. The genome similarity is around 70% in zebrafish compared to the humans. And this organic structure is very similar to the humans. So the genetic manipulation, for example, the microinjection to the fertilized embryos, is very easy, and drug screening is also convenient to perform. So these characteristics increase the zebrafish is very popular as a new zebrafish disease model. This is the swimming zebrafish, and this is under the UV light. We can see this is transgenic lines, the EGFP specific transgenic lines. And this is the vascular specific EGFP transgenic line. We can see the zebrafish vascular system under a fluorescence microscope. So in our lab, we have developed several zebrafish models for human chronic diseases. For example, this is the zebrafish model for obesity. I will introduce the details later. And the zebrafish model for type 2 diabetes mellitus. This is the characteristics of this diabetes model include hypoglycemia, glucose intolerance, insulin resistance, responsive to anti-diabetic drugs, and they share common pathological pathways with human diabetes. This is the zebrafish model for diabetic nephropathy. I'm sorry, because this is unpublished data, I cannot show the details. We just developed as a specific transgenic line that we can track the proteinuria of the diabetes zebrafish. So we can find this the continually increased proteinuria leakage of the diabetic nephropathy model. This is the zebrafish model for osteoporosis. Since the zebrafish scales have a similar structure and function to mammalian lamellar bone, which contains both osteoblasts and osteoclasts. As observed in humans, we can use the glucocorticoids to induce osteoporosis using zebrafish. These are the zebrafish scales that stimmed with trap to detect the osteoclast activity. We can use this model for the drug screening to find the drugs or natural products that have anti-osteoporosis activity function. We also have the zebrafish model for cancer xenotransplantation. This is also a convenient tool for anti-cancer drug screening. Among these models, today I will focus on the zebrafish obesity models and introduce its application. So what is obesity? Obesity is now a global epidemic. The overweight and obesity are defined as abnormal or excessive fat accumulation that presents a risk to health. As we all know, the body mass index is a simple index of weight for height that is commonly used to classify overweight and obesity in adults. This is the weight categories based on BMI. We can see that healthy weight is among the 19.5 to 24.9. Overweight is defined between 25 to 29.9, and obesity is defined as 30 to 39.9, and over 40 is defined as severe obesity. The cause of overweight and obesity are increased intake of energy-dense foods that are high in fat, or an increase in physical inactivity. The health risks of overweight and obesity are cardiovascular diseases, diabetes, musculoskeletal disorders, cancers, and so on. This is the facts about overweight and obesity. In 2016, the world ranking for the high risk prevalence of obesity is 36.2% in the United States and 35.4% in Saudi Arabia. Japan has the lowest prevalence rate of obesity in developed countries, which is just 4.3%. The prevalence of obesity named among adults increased rapidly in the past 40 years. Obesity is one of today's most blatantly visible public health problems. It's a big challenge for the researchers to prevent the prevalence of obesity or treat this complex condition. So as I said, as I introduced previously, zebrafish is a useful model animal for human disease. So how should we do to treat this disease? Firstly, we need an obesity zebrafish model. We have developed two types of obesity models of zebrafish. First, I'd like to introduce how to develop an obese model using zebrafish larvae. As previously reported, researchers have developed a zebrafish obesogenic test for screening molecules target adiposity in 2011. This is the protocol for the ZO test. It just takes 3 days. On the first day, the zebrafish larvae were fed with a high-fat diet or standard diet, and then followed by 1 day of starvation to empty the lumen. On the third day, expose the larvae in the test material and solution. The Nile rat and the adipocyte can be observed under a fluorescence microscopy. So the Nile rat staining performed before and after the test material exposure to calculate the adipocyte tissue reduction rate. Based on this reference protocol, we made a minor arrangement that shortened the experiment period to 2 days. First day for high-fat diet treatment, and the second day for test material exposure. Nile rat was also performed before and after the exposure. You can see the visceral adipose tissue can visualize under the fluorescence microscope. So this modified protocol is also effective and demonstrated that the ZO test is a whole organism alternative testing assay for obesogenic and anti-obesogenic compounds and mixtures. Next, I will show you how to develop an obese model using adult zebrafish. There are 3 approaches to develop an obesity model, including induced models, transgenic lines, and the mutant lines. The simplest and the closest to the human obesity etiology approach is overnutrition. This report is our obesity model published in 2010. We created an obesity model by overfeeding Altemia, which is an easily accessible food and is normally used in zebrafish facilities worldwide. For the details, we divided adult zebrafish to normal feeding and overfeeding groups. Normal feeding groups were fed once daily with 5 milligrams of Altemia per fish, and the overfeeding zebrafish were fed 3 times daily with a total of 60 milligrams of Altemia. So after 8 weeks of overfeeding, the zebrafish got significantly increased body weight and plasma triglycerides compared to the normal feeding group. And also the So the intra-abdominal adipose tissue can be visualized in a live wing zebrafish under a fluorescence microscope by Nile rat staining. After 4 weeks of overfeeding, the adipose tissue was significantly increased and reached the maximum after 8 weeks of overfeeding. Since the treatment of obesity is diet and the practice, we tested if the diet can function as to treat with this obesity. So we can say 2 weeks of calorie restriction was effective to decrease the adipose tissues. These are the 3D micro-CT analysis. So the gray colors indicates bone, and the yellow color indicates the adipose tissues. These are the 2D images after the software analysis. The orange color indicates the subcutaneous adipose tissue, and the yellow color indicates the visceral adipose tissue. We can say that after overfeeding, the total body fat and visceral adipose tissue and subcutaneous adipose tissue were significantly increased. This is the oil red O staining of frozen sections of the zebrafish liver tissue. So we can say the white arrows indicate the lipid droplets. The transcriptome analysis of the visceral adipose tissue revealed that obese zebrafish and humans share common pathological pathways related to the lipid metabolism. So we can conclude that this obesity zebrafish model is a go-to to validate the anti-obesity effects of natural products for their potential use in the treatment of human obesity. So as the applications, we use this obesity model to evaluate the anti-obesity effects of numerous natural products. For example, the tomato, the green tea, lemon, and use this as one of the citrus fruits origin in the Asia. And the cereals, the green cereals and the brown cereals. And this is origin in Japan, one of the citrus fruits, and the cauliflower fungus. We also identified several molecules targets related adiposity using this obesity model. So now we have two obesity models using zebrafish larvae and adult zebrafish. So if we have, we can say in the for one sample's evaluation of the anti-obesity functions, we use the adult zebrafish. We will spend 3 or 4 weeks. So if we have a library of the test samples, so how we can do it? We will spend money and times. So we developed a systemic technology to evaluate the anti-obesity screenings of the natural products. We just use the zebrafish larvae as a first screening, and then validated the selected test materials with the adult zebrafish. This is the we performed a small-scale screening of 42 natural products by the zero test. The zebrafish larvae, the red fish is the zebrafish larvae, were put into the 6-well plate and exposed in the ethanol extraction of test materials. After the Nile red staining and the validation of the adipose tissue volume, we can get this conclusion. Among the 42 natural products, 6 natural products were found that have 50% or more visceral fat reduction effect. So among the 6 test materials, I will show you the result of the, this is the green tea extract. As it is well known that green tea is one of the most famous and popular beverages worldwide, and the green tea riches in polyphenols such as catechins and flavonoids. These are the chemical structures of the green tea catechins. Among these catechins, the EGCG is the major one in green tea and has demonstrated the high-risk biological activity among the green tea catechins. So EGCG has multiple effects on human pathological and physiological processes, including antioxidant, anti-cancer, anti-inflammatory, anti-fibrosis, and so on. So this is the result of our ZO test of GTE. The GTE is green tea extract. You can see that 1 day of starvation induced a decrease in the VAT volume in the control group. This is the before and this is the after. And in the GTE treated zebrafish larvae, the volume of VAT has showed a more remarkable was quantified by a neo-red fluorescence signal. We can say that approximately 70% reduction. So we then concluded that GTE decreased VAT volume induced by a high-fat diet in zebrafish larvae. Next, we orally administered GTE to adult zebrafish. This is the protocol of the experiment design. GTE was pre-treated for 2 weeks, followed by 1 week of overfeeding. The short term of overfeeding increased the body weight of overfeeding group compared to the control group, and the GTE treatment showed a little reduced body weight gain. But GTE significantly reduced the VAT volume compared with the overfeeding group, and also the plasma triglyceride and plasma total cholesterol are also significantly decreased in the GTE treatment group. So we can conclude that pre-treatment of GTE before the induction of obesity could prevent its development, suggesting that daily consumption of green tea prevents an increase of in the VAT and promotes the health. To investigate the underlying molecular mechanisms of GTE, we conducted transcriptome analysis of liver tissues of from the adult zebrafish treated with or without GTE, and then perform the pathway analysis. We can see in the Wnt canonical signaling pathway, the Wnt beta the Wnt freezed complex and its downstream genes GSK3B and CTNB1 were upregulated by GTE. The qPCR analysis revealed that GTE significantly upregulated the GSK3B and showed a tendency to active CTNBB1 expression. This result indicates that GTE ameliorates obese in adult zebrafish might be an active Wnt beta-catenin signaling pathway. And in addition, in the AMPK signaling pathway, the major downstream pathways of AMPK, such as the SREBF1 that related to lipogenesis were downregulated by GTE. And the later qPCR revealed that the messenger RNA expression level of the PRKA1, which is the AMP subunit 1, and the AMP subunit 2, this showed a tendency towards an increase. This is showed an significantly upregulated by GTE treatment. On the contrary, the expression of SREBP1 showed was significantly downregulated by GTE, which is consistent with this result. We then concluded that GTE ameliorates obese in adult zebrafish might via active AMPK signaling pathway. Finally, we compare the molecular mechanisms between zebrafish and the mouse that are treated with GTE. Most of the genes showed similar alterations in the gene expression between zebrafish and the mouse. This data revealed that zebrafish treated with GTE may share common molecular response with mammal and further demonstrated the utility of this animal model. So in summary, we developed the obesity models using love and adult zebrafish. The love obesity zebrafish model is suited for a high throughput natural product screening as first screening, and the adult obesity zebrafish model is benefit for the validation of the effects of the selected natural product. So I would like to appreciate Professor Nishimura and Dr. Shimada and all the other lab members. I also thank our collaborators and the funding supporting this work. Thank you for paying attention.